I'm Dr. Monty Pal from City of Hope, and this is On the Edge of Breakthrough, Voices of Cancer Research. Each episode, we bring you the minds behind the science, the stories behind the data, and the breakthroughs that could change everything. Let's dive in. Welcome everyone to On the Edge of Breakthrough, Voices of Cancer Research. I am super excited today to have my dear friend and colleague, Dr. Tanya Dorff, on the program. She is the Vice Chair of Clinical Affairs for the Department of Medical Oncology here at City of Hope, and she's also the Director of GU Oncology, which actually makes her my boss. So welcome, boss. It's great to have you on the program.
Good to be here.
Well, we've known each other for eons. So just for the audience, we were overlapping in medical school at UCLA, although we didn't quite get to know each other way back then. But, I wanted to kind of hear a little bit about your origin story. What was it that kind of drew you into medicine first and foremost?
So I knew I wanted to be a doctor from the time I was 3. It was just what I wanted to do with my life was help people. And it felt like that was a way to be helpful in the world, was to heal.
And is that rooted in family members that were physicians, or how did that kind of come about?
I have wondered how it came about. I think maybe my female pediatrician was a role model.
Got it. Yeah. And I will say that you and I were at UCLA at such an interesting time as it pertains to oncology, right? I mean, we're seeing targeted therapies evolve for the first time. A lot of it kind of built on the backbone of technologies developed here at City of Hope. What was it that took you further from the field of medicine into oncology?
So really it was the clinical experiences, just being with people in this time of great vulnerability and being able to help them navigate a very complicated disease, being there together with their whole family and support system. And then also the scientific interest. But probably what really cemented my interest in oncology was my mom's diagnosis of APL when I was 18. A disease that was uniformly fatal until the understanding of the chromosomal rearrangement led to the development of the all-trans retinoic acid. So she still got standard chemo because that's what they were using back then. But then she got the all-trans retinoic acid. They didn't really know about retinoic acid syndrome. She was in the hospital for 6 months, very sick. But she's still with me today. She was cured. So I think that beautiful story where if you can understand the biology of the disease, you can take something that's fatal and turn it into curable was a really compelling reason to go into oncology, and even more so made me really interested in clinical trials and clinical research because I could see the difference that it made to people's lives.
That's just fascinating. And I remember in the distant past, you and I have actually discussed your mom's story, which I think is just so heartwarming, and I'm so thrilled that she's still with us today. Did you have a lot of interactions with her oncologists back then? I'm curious to know if you had, what your interface was like with the medical system back then.
Yeah, I did not have the kind of interaction that I see my patients' children having with me today. I guess it was a different time. He was a spitfire. He would come in and yell and throw papers around, and it was much more the older, I'm going to tell you what we're doing, and then a question and answer kind of a discussion relationship.
Got it. I think I remember that time training at the same, sort of phase. I do recall that the dynamics have definitely changed in the doctor-patient interaction, right?
For sure.
Yeah. Now, so we separated after UCLA for medical school. You went to New York for residency and then came back to USC to do your fellowship. And I'm curious, I believe it was that time that you got sort of inspired to move into the field that we're both in right now, which is genitourinary oncology. And just for the audience, that's the field assessing prostate, bladder, kidney cancer. Is that right?
Yeah, it was really my mentor who I connected with during fellowship, Jacek Pinsky, who ran a prostate cancer lab. And so, between working on grants with him and clinical trials and working in his lab together with Sia Donishmond, we spent many hours slicing up prostate tissue and staining it and looking at it under the microscope. By the end of fellowship, it was just, that was what I knew the most about. And it was also a really nice disease for me, being someone who wanted to have long-term relationships with my patients. So I got to know these men for many, many years longitudinally as prostate cancer patients.
Oh, that's a really great point. And it's funny for me to think about these two luminaries like yourself and Dr. Danishmand, who's a terrific surgeon at USC now, focused primarily on bladder cancer, sitting there at a microtome, sectioning specimens, right, all those years ago. It's a real lesson in how small this world of oncology is, wouldn't you say?
Yeah.
Well, tell me a little bit about, what came after that. So you've obviously forged an international reputation in prostate cancer research. Tell us about kind of the early days that sort of inspired you to sort of take this path within prostate cancer. You spoke about the bench work. How did that translate to clinical work for you?
My big interest was in immunotherapy. From the get-go, it just made sense to me that if our immune systems could identify the cancer and engage, that could lead to really good remissions. But there wasn't a lot of immunotherapy happening at USC at the time that I was beginning my career. So, we did participate in the rollout of Sipuleucel-T, which was very exciting. It was one of the first immunotherapies available for any cancer, and certainly for prostate cancer. And there was huge demand. Also around that time, abiraterone and enzalutamide were in clinical trials, and seeing how my patients did so much better with this next generation of androgen receptor targeted therapy was just so rewarding and also horrible. Like, while in that black box period between the representation of the phase 3 as being positive and seeing all these patients on trial doing so well, and then those group of patients who were waiting for the drug to get approved and couldn't access until it did get approved. So, it was a roller coaster of emotions during that time, but that made a really big difference. And actually now, after the availability of drugs like Abi and Enza, we're seeing a little bit more of neuroendocrine transformation of prostate cancer, in part because patients live longer and the cancer just has more time to evolve, not because of anything bad about those drugs, right? Patients definitely live longer and better. But that harkens back to the work that I was doing in Jacek Pinsky's lab. So a very long answer to your question, but we were looking at neuroendocrine differentiation in primary prostate tissue among patients from different ethnic backgrounds. And also Jacek was very interested in the role of IL-6 in transforming a normal prostate cancer cell or a normal prostate cell into a neuroendocrine cell.
Got it. And, for the audience, this neuroendocrine differentiation, it's this really sort of aggressive sort of phenotype that prostate cancer takes on, through selective pressure, perhaps, maybe innately in some cases. Brilliant to have that sort of focus. But before we dive into some of those rarer subtypes, I wanted to kind of get to your transition over to City of Hope, because I think this is really where you've really made such a huge mark on the field in terms of immunotherapy and its application for prostate cancer. If I may, I'm going to share with the audience a little bit of that transition story. So this is, how many years ago now? It's about 10 years ago, Tanya?
Almost 8.
Almost 8 years. Okay, got it. So I vividly remember the time at which my former partner here at City of Hope, who focused on prostate cancer, announced that he was leaving. And it was a really tough moment because at the time, the program was essentially just me and him, right? And so I remember my chairman at the time coming to me and saying, who would you like to come in and take over this position? And I said, I really want to bring in a leader in the field, somebody who could take the helm of the program. And I have one single name in mind, and it's Tanya Dorff. And I remember my phone call to you actually came at a very awkward time. I love telling this story. I was in the back seat of the car of another, prostate cancer legend, Neeraj Agarwal, one of my very close friends in the field. And so I called you up. I said, Tanya, how would you like to come and lead our GU program at City of Hope and be my boss? And I remember he turned around to me and said, Monty, what am I, chopped liver? So tell me about, that experience and what sort of drove you to maybe jump ship and come over to City of Hope.
Well, absolutely. It was a huge draw to be able to work with you. I'm such a huge fan of yours. You're just one of the most brilliant and kind and generous and productive physicians and human beings on the planet. So the opportunity to work with you was definitely a big draw. But also it was because there was CAR T-cell therapy starting up here at City of Hope. Saul Priceman had developed a PSCA targeted CAR T-cell, and the opportunity to lead a phase 1 first-in-human trial with a treatment that I thought really had promise was too good to pass up.
I've actually been witness to this amazing sort of process through which I think this was just like, sort of at the level of informal discussions, when you'd come on board. And I think Saul maybe already had some sense of what he wanted to develop preclinically, but I've seen it really go. We always talk about bench to bedside. We talked about it a lot on this podcast, but this is one of the best examples I can think of bench to bedside. Now you have a first author manuscript in Nature Medicine that reflects the data, one of the most prominent journals in our field. Can you kind of walk our audience through how you went from looking at a CAR T cell, just theoretically, and turning it into something that's actually moving into patients?
Yeah, so it's very hard to take things that work in the lab and translate them into human patients. But Saul had done a lot of the preliminary work, the IND-enabling studies, and he has a really good mouse model, as good as one can get. Particularly immunotherapy is very challenging because you have to have an immune system. And most of the time when we're having human cancers in mice, we have to suppress the immune system, right? But he developed an immune competent humanized PSCA knock-in mouse model that allowed him to do really robust and rigorous experiments with the PSCA targeted CAR T cell, showing that it honed to bone metastases, which is really important since most of our patients have bony metastatic disease, and that it could activate and proliferate and do what we needed it to do. So putting that into a human being, we had to think about whether we wanted to give a big dose or a little dose. Obviously we don't want to enroll a patient and give them a dose that's really not going to be able to help them. And yet we weren't sure how active the cells would be, how much toxicity. We were looking at the hematologic malignancies and of course trying to learn from what doses they had used, what doses were effective. We also had a big question about whether you needed that special preparatory chemo, the lymphodepletion. It makes a lot of sense in heme malignancies, but we weren't sure whether it was going to be necessary for solid tumors. So thinking about the design of the phase 1, we went back and forth a lot, and it really was bench to bedside and bedside to bench. All throughout the process, we were looking at what was happening in the patients, trying to replicate it in the lab so that with the next iteration, we could modify and do better.
Yeah, I just want to emphasize that bench to bedside back and forth that happened in the process. Even with the phase 1 clinical trial, my understanding is that, and I saw this in your Nature Medicine paper, it was embedded with really kind of heavy correlates that might inform future studies. Maybe you could tell our audience a little bit about that.
Yeah, I don't know how deep science we want to get on the podcast, but certainly we were doing biopsies before and after so that we could look at what was happening in the cancer microenvironment before the cells went in. And then afterwards to see whether the CAR T cells actually got in there, whether the cancer cells were dead, and what else was happening, whether the expression of the antigen we were targeting was lost. We had a lot of questions for those tissue samples. And then we're also partnering with the Kuhn Center for any rare cell events get captured. And we're continuing to do a lot of work there looking at PSCA, PSMA expression on circulating tumor cells before and after. We were able to look at bone marrow circulating tumor cells versus peripheral, and we found tremendous differences there. And this kind of really interesting push that the lymphodepletion seemed to push the cells out into the periphery, and then the CAR T cells eliminated them. So yeah, really a lot of deep science. We also wanted to see T cell receptor repertoire expansion and diversity. So really trying to learn as much as we could from every patient so that when we saw some good responses and some patients who didn't respond, we could try to start to understand what to do different or which patients might be the best patients to enroll in future trials.
Absolutely. And, I've had a lot of discussions with Rose Lee, who's one of our, I think real rising stars here at City of Hope on our radiation oncology group. And correct me if I'm wrong, I think that she is working with you now on some hypotheses around using radiation treatment to maybe stimulate the CAR T response. Is that right?
Yeah, so we submitted an R01, which got scored 7th percentile.
Congratulations, awesome.
Looking at the role of radiation in priming the tumor microenvironment. So When we did the phase 1 trial with the PSA-targeted CAR T cells, as I mentioned before, we wanted to see whether the lymphodepletion was necessary. And we got very minimal CAR T cell expansion. We did see some PSA go down, which was interesting. So the cells alone were doing something, but they didn't have that big expansion that we needed. And so Saul's postdoc went back and looked at what the lymphodepletion was doing in the tumor microenvironment and showed that it was having these favorable changes, increasing antigen presentation and dendritic cells and reducing the suppressive kind of macrophage component. And then another postdoc started working on the role of radiation and what it could do in preconditioning the tumor microenvironment. So yes, that's what we want to put in now to the phase 1b, because even with the lymphodepletion, we had some patients with good expansion, but not adequate persistence. So the responses we saw in the prostate cancer patients waned with time. And it was in part because the CAR T cells just weren't continuing to make themselves. And we think some of that has to do with the suppressive microenvironment. So yes, the next generation is to look at radiation, and then lymphodepletion, and then the CAR T cells.
Honestly, Tanya, as we talk about all this amazing work you're doing in the CAR T space, I'd be remiss not to really sort of acknowledge the different pathways for funding. I mean, obviously, you've been very successful when it comes to grantsmanship. Congrats on the R01 score and all that great stuff. But I think philanthropy also plays a big role in us being able to do some of these novel things, wouldn't you say?
Absolutely. I mean, having those philanthropy funds allows us to fill the gaps and do some more preliminary research that helps us get the grants. And in my case, it's actually helping keep one of my clinical trials up and running, thanks to these generous donors, Kilgallen and Zamaripa. Without that generosity, we would have really struggled to keep the CAR T cell trial going. So
fascinating. I wanted you to give us some perspective on what it's like to be a solid tumor investigator and in that CAR T space. I get the sense that that's one of the unique things about City of Hope. We actually had Steve Foreman on the program a couple months ago, in fact, and he gave a wonderful distillation of, the role of CAR T across tumor types. But, one thing that we didn't really allude to, maybe because of his humility, is what a great opportunity we have as solid tumor investigators to kind of get into that CAR T space. So maybe you could tell us about some of that City of Hope secret sauce around our CAR T program.
Yeah, absolutely. I would not have felt comfortable doing something so new and different and innovative and intense without having the mentorship of someone like Steve Foreman, who thinks so deeply and has such broad experience and knowledge. So having him at the helm was critical. But beyond that, there's also an amazing team of pharmacists and nurses, the manufacturing team that produces the CAR T cells, the regulatory that interfaces with FDA. It takes a huge team to do something like this, to take a scientific idea, a scientific design product, all the way through to human trials in a way that's ethical and as effective and efficient as possible. So I think that's the secret sauce, is really that we have a very big team, and there's just a really great sense of collegiality and collaboration, right? So the heme folks, the immune effector cell sort of heme rounding team, very kindly, takes primary management of the patients when they're in the hospital. Although, we certainly see the patients every single day and try to provide guidance based on the specific types of reactions that we see in our prostate cancer patients that might be a little different than what they see in heme malignancy patients getting CAR T cell therapy.
Totally. And you being my academic big sister, I oftentimes follow in your footsteps in a lot of things. And I started doing CAR T stuff after you did, in fact, mainly in the context of renal cell. And I have to vouch for what you said there. I mean, it's just so amazing to see the level of care that patients get here when they're getting CAR Ts and some of these novel immune-based therapies. The level of oversight that our hematology team offers in collaboration with us is just, it's bar none. It's really amazing.
Right. And all that help and experience and wisdom and infrastructure, I was then able to apply it when we were looking at doing bispecific T-cell engagers, where we knew we were going to see some similar types of immunotherapy side effects. We were able to do that outside of the heme ward, the CAR T ward, and do it through really the dedication of some of our research nurses in mentoring and educating the floor nurses, and the dedication of those floor nurses to calling me directly if there was cytokine release happening. So I think every time I looked a patient in the eye all along the way for any of these new phase 1 trials, I had that confidence to say, we're going to take care of you. Like, this is a new, exciting thing. It's a little bit scary, but we're going to get you through this. And we really believe this may help.
Indeed, indeed. We've done a look at our audience, and we do have, a certain subset of physicians, investigators, et cetera, listening in. We have a lot of patients listening in. So just very briefly, this whole phenomenon of bispecific T cell engagers, the way that I would usually explain it to a patient, and tell me if this is accurate, is you've got one side of the molecule, right, that tethers to the immune cell. The other side of the molecule tethers to the cancer cell. And you're trying to bring the two in kind of close proximity to one another just to really elicit a profound immune reaction. And that's why, when Dr. Dorff mentions, for instance, all these, sort of complex reactions that may occur, they really require such tight supervision and oversight in the hospital. But let's shift to that, because I did want to bring up these bispecific therapies. And again, here's a place where I think you've really established yourself as an international leader. We've had a number of programs, some of which we've had first patient in this bispecific space. Tell us a little bit about the stuff that you're most proud of coming out of our bispecific arena.
Well, I think the partnership with industry is something I'm really proud of. Industry has recognized City of Hope's ability and commitment to getting clinical trials open and accruing patients and taking good care of the patients that we accrue and having the right infrastructure. So, I think it's so wonderful to be able to talk to industry and say, this is how I think you should do the phase 1. And all along the way, there's a whole group of investigators, everyone that's enrolling at different sites, providing feedback as we're accruing patients. But I think being able to talk even before and look at the preclinical data and provide feedback as to what more we think we would want to see before we feel comfortable opening the trial and supporting it has been really, really a fun and engaging and satisfying process. But with the bispecifics, we really had to set the learning curve for how to manage some of the toxicities we were seeing, how to recognize them, how to define them, how to best treat them, and then moving into prophylaxis so that patients didn't have to go through the side effects once we knew timing and what different strategies might work to mitigate. So we also partnered a bit with industry, thinking about how to move such a powerful and exciting but potentially toxic treatment out into community oncology where most patients get their care. So we did start to have some conversations about what infrastructure would be needed in a community oncology clinic. What types of resources could make that feasible if the bispecific was going to have a fairly high rate of immune side effects like cytokine release syndrome.
Yeah, it's interesting. I just wanted to give a quick shout out to one of our investigators, John Williams, here on campus. He gave just the most stellar talk. We talked about bispecifics here, but actually trispecifics, these molecules that can now hinge onto multiple entities within the sort of cancer milieu, and with that better engagement elicit these really wild responses. So I'm really looking forward to seeing a lot of our internal development. You already alluded to it in the context of CAR T cells. We have it going on in terms of bispecifics and trispecifics now. It's just such an exciting time to be here, isn't it?
It really is. It's there's many more opportunities to develop compounds that are really going to make a significant impact and not just be incremental advances.
Yeah, indeed. So, we were just chatting ahead of the show today about your various roles here on campus. You've taken on a role not just as the head of our GU group, but also the vice chair of clinical affairs. And, I just wanted to kind of get a flavor for what you think the possibilities are. We have this huge expansion, which has all happened right before our eyes during the time that we've been here with our Irvine site opening up. We now have sites in Chicago and Atlanta and Phoenix that have opened their doors and are seeing lots of patients. What do you think we can do, maybe not just from a clinical standpoint, but from a research standpoint, to harness that growth that we've had?
Well, I think it speaks to City of Hope's commitment to being accessible to more cancer patients, because we are proud of the very high level care we provide. And so having these centers will allow more patients the possibility of coming to one of our sites. I think if we're really going to realize that promise, we want our sites across the country to be able to run some of these innovative therapeutic trials like CAR T-cell therapy and bispecific T-cell engagers. And we're just going to have to learn how to work closely from so far away, and what that looks like to have supervision and mentorship across state lines. But we've learned, through COVID, for better or worse, that we can do a lot by Zoom, right? And I think we will have better diversity in our accruals by having patients accruing at different sites. And we'll be able to mentor, significantly more next generation leaders of clinical therapeutic development.
Indeed. You know how strongly I feel about mentoring. I think you're a terrific mentor yourself. What is your advice to folks that are entering into the field of oncology and maybe feel like they might have a penchant for academic medicine?
Well, I encourage them to pursue it. We need great minds to come and join us in this work that is so fulfilling. Since I came to City of Hope, I have to say there have been so many days when I'm driving home and I think I did exactly what I want to be doing today.
Oh, that's cool.
Whether it's a day where I've touched some patients who are on clinical trials and they're doing really well, or whether it's a day where we learned something from the lab that's going to help us in our next clinical trial. It's been a wonderful, wonderful part of my career. And so I think it is accessible. I was told that women with children shouldn't go into academic medicine, but I'm glad I didn't listen. And I think that mindset is more a thing of the past. It's still a big commitment. You have to work hard to make progress and become, sort of an expert in your field. But it is doable while raising a family, and you're certainly a very good example of that. I see all the time you're teaching science to your kids and their friends. And
Tiger dad. Yeah.
So it is doable, and I think people should not be turned off by the thinking that it's just too much. It's like 2 or 3 jobs in one, which it sometimes feels like, but it is doable and so worthwhile.
Yeah, I want to dig deeper on something that you said that it really kind of troubles me, which is that, you heard at some point in your career, and you shared this with me before, that, women shouldn't go into academic medicine. And, I believe that's, a huge fallacy, right? I know you feel the same way. But I think it's a message that still comes to, some folks who are in training. What do we do to kind of combat this? What's our way around it?
Well, I think we have to meet people where they're at. I think timelines for academic promotion and milestones have to take into account that there are times in our lives where we need to focus a little bit more on family and what's going on at home, and times where we have more hours and more bandwidth to devote to the clinic and to our academic research. And I do think many universities have taken that into account and give a little bit of extra runway to young folks starting out in their academic careers who are also starting a family.
Indeed. I mean, I think you're such a powerful example of what, folks, can accomplish in academic medicine, irrespective of gender. I mean, I've always looked up to you that way. I love, the interactions that I've had with your kids. Your daughter's at Hopkins now. And by the way, for the audience, she is just a carbon copy of Dr. Tanya Dorff. I mean, you look more like sisters than mother and daughter, to be honest.
She's better than me in every way, I like to say, and that there's nothing that makes me happier.
But my understanding is that she's an aspiring GU oncologist. Is that right?
She's going to be a urologist. It's just written in the stars. Yeah. She's had some really good interactions with our lovely colleagues, Clayton Lau and Kevin Chan. And she's working in some urology research labs. So she worked in Dave McConkey's lab. And so, I think she's well on her way. Oh,
that's very cool. So, Tanya, we've talked about business and more business and more business, but clearly, there's some cool things that you do outside of the workplace that help you unwind. If you're willing, I'd love for you to share with the audience one of your favorite hobbies, which I'm aware of, they may not be aware of. Go for it.
Well, so I sing in a Lithuanian choir. Last year, it was a women's choir with women from San Francisco and Los Angeles coming together to perform in a festival in Vilnius in Lithuania. 11,000 people singing. It was one of the most moving experiences of my life. And now I sing in a mixed choir and will be performing this weekend at LT Days and performed in Cleveland at the Song Festival. But I have to say, singing, all of you should give it a try. You know, when you've had a tough day at work, if you put on music and start singing, it just really lifts your spirits. And I think it's fun to challenge yourself learning harmonies and new melodies and new rhythms as well.
Now, you did share with me that in the mixed choir, the men are kind of holding you back a little bit. Am I right?
Not at all. I love the men's voices.
You know, we always tend to sort of wrap up the show by asking a tough question. It's having to do with the title of this program, which is On the Edge of Breakthrough, Voices in Cancer Research. We always ask our guests, what does On the Edge of Breakthrough mean to you?
I think it means taking those leaps, right? Seeing what's happening in the lab, seeing what's happening in the clinic, and connecting them in a way that will lead to advances that are big leaps. So we're blessed to have both robust grant funding and philanthropic funding and partnerships with industry that allow us to do that, because to do something safe and tried and true, okay, you know, you'll get papers, you'll find some good treatments, but to really find something that is completely revolutionary, you have to see the landscape, you have to have access to really great basic science and innovative new treatment and therapeutic development, and you have to be able to put it together. And I think that's something that City of Hope does really well.
Brilliant. Well, there you have it, guys. What a wonderful conversation today with you, Tanya. Thanks so much for joining us on the program. And I'm sure we're going to have you back in years to come to really sort of discuss where the field of immunotherapy and prostate cancer sits.
Thanks so much, Monty.
Thanks for tuning in to On the Edge of Breakthrough. See you next time for more insights from the front lines of cancer research and care.